If you look at the forehead of a striped cat, right about the eyes, you will almost always find a distinct M-shaped marking. This feature is so common that the orange tabby is often mistaken for a specific independent breed of cat. In reality, these markings appear across wildly different pedigrees. The Maine Coon, for example, displays the exact same stripes and forehead signature, despite its unique size and coat length. Tabby markings represent a primordial genetic blueprint found across the feline lineage, existing independently of any single breed. To understand how this blueprint functions, we have to look past the visible coat and examine the microscopic structure of a single hair. This is an agouti hair. Individual follicles contain alternating bands of light and dark pigment rather than one solid color. When millions of these hairs grow in proximity, their pigmented segments align, creating the visual effect of a solid dark stripe on the body. The stripes and whorls we see are actually the result of highly synchronized biological printing occurring at the microscopic level. While the agouti hair is the base building block, it assembles into 4 distinct macroscopic patterns. The mackerel pattern creates the recognizable vertical tiger stripe layout. The classic pattern produces thick, swirling bullseye shapes along the cat's sides. In spotted tabbies, these stripes break into disconnected dots. The ticked pattern is more subtle. The stripes are restricted to the face and legs, while the rest of the body appears solid. Felines can even display complex combinations, such as the tabbyco, where tabby stripes overlay the orange and black patches of a calico. These visual variations, from swirling bullseyes to disconnected spots, all originate from the same underlying genetic instructions. This physical alignment is driven by cellular architects that begin the printing process before the cat is even born. During fetal development, the Taqpep gene establishes an invisible pre-pattern in the skin, acting as a biological grid defining the future coat. A specific mutation can alter these boundaries, stretching uniform stripes into the swirling blotches seen in classic tabbies. Taqpep sets the borders, but a secondary protein called endothelin-3 serves as the ink, populating the dark regions defined by the pre-pattern. This 2-stage signaling mechanism ensures that dark and light pigments occupy their assigned zones, creating perfectly localized color differences. A cat's coat is the product of a 2-stage developmental mechanism that translates genetic instructions into physical patterns at the cellular level. This biological printing process is foundational to feline DNA, existing in the vast majority of domestic cats. Many cats appear solid-colored because of epistasis, a genetic phenomenon where a dominant gene masks the expression of the underlying pattern. This means a solid black cat often carries a complete, functional tabby blueprint that is simply hidden beneath its dark coat. Direct, harsh sunlight can sometimes bypass this masking effect, revealing faint ghost stripes on an otherwise solid black cat. The species preserves this foundational coat regardless of the cat's visible color, maintaining the tabby blueprint throughout its history. Long before we could sequence these genes, human cultures recognized and celebrated this intricate visual pattern. The word tabby itself traces back to the Al-Atabiyya quarter of Baghdad. This region was the center of production for a luxurious, wavy silk known as a tabby. When this silk reached the West, its moire patterns were compared to the cat's coat, lending the fabric's name to the animal itself. While biology explains the Taqpep gene, ancient legends offer different origins for the forehead M, attributing it to figures like Mary, the Prophet Muhammad, or the sacred cats of Egypt. The domestic tabby is a living record of this ancient evolutionary signature, carrying a complex biological history from the wild into the modern home.